Hello, everyone. My name is Christopher Rose. I'm the Director of Discovery Proteomics at Genentech within the Microchemistry, Proteomics, Lipidomics, plus Next Generation Sequencing group. And I'm very excited to talk to you today as part of this TMT Symposium, the third annual, about some of the work that we've been doing trying to increase the throughput of multiplexed proteome analysis through intelligent data acquisition. Now, first, I'm going to touch on a couple different topics today, but one of them is single-cell proteomics. And I've been starting my talks nowadays by putting a plug in for this review that we had as part of the Nature Methods special issue on proteomics earlier this year. And within this review, we actually combined forces within our group, which is really within the proteomics wing of the department, as well as the next generation sequencing group and some of the folks in that department. And we kind of put together our thoughts on single cell proteomics and how we both attack it from two different angles. Obviously, for our group, we're typically using mass spectrometry as the readout for single cell proteomics measurements. The next generation sequencing group uses sequencing mediated through antibody binding and does that to measure protein levels through things like site-seq, and we even touched on things like MIBI within this review. So I think if you're interested, you came to this talk interested in single-cell proteomics, this is a nice place to start to try to understand how different fields are actually coming at this problem from two different angles. So today I will be talking mostly about an intelligent data acquisition program or approach that we're calling PairQuant. And we're using that on two different applications. We're going to be using it on cysteine profiling, and then we're also going to be using it on single-cell proteomics. Now, at the heart of this approach is really the hyperplexing methodology. So I'll introduce hyperplexing, why we think that this is an attractive approach for these types of analyses, and then go into how we've actually addressed some of the shortcomings of hyperplexing through intelligent data acquisition. Now, for these two projects, we have two different teams. So for the cysteine profiling, this was really led by Hannah Budieva and Taylor Ma and Mina Choi within the chemoproteomics group and the computational proteomics group, respectively. And then for the single cell analysis, this work was done in lab by Tommy Chung and also helped out by with Ying Zhu in terms of some of the sample preparation techniques that we employed. So I'm going to start out talking about cysteine profiling. This is kind of the test bed that we had for some of these methodologies I'll be discussing. And then after that, we'll go into some single cell proteomics. So firstly, what is cysteine profiling, within the realm of chemoproteomics? So generally, when we're talking about cysteine profiling, we're trying to understand which cysteines on a protein are going to be reactive with a particular chemical or, say, an inhibitor or therapeutic that we're trying to develop. And cysteines themselves can be attractive covalent targets within proteins. Some of them actually exist within the active sites, and active sites have traditionally been nice places to put therapeutic molecules because they generally have a good binding structure. And when cysteines are within the active site, it actually allows you to kind of anchor those in and to lock them into place, effectively inhibiting the protein. And in this case, it might be something like a kinase, like BTK, but you could have other proteins as well. Now, when we're thinking about expanding that and moving past active site binders or proteins that are classically druggable, we're going to be talking about how do we start to drug the undruggable. And kind of a revolution in this space was really around KRAS G12C, which had been thought to be undruggable, but had been shown through some really nice experiments that you can actually get molecules to bind. They may not bind too tightly within this particular pocket in KRAS G12C, but there is this covalent residue, right, the cysteine that is actually the mutation, where you can lock it into place through covalent modification. And one of these molecules has already been approved. There's several other in the clinic. And this approach has really kind of revolutionized the way people think about trying to attack proteins that may not have classically been druggable, and has raised the question which cysteines on proteins within the proteome are actually able to be modified. So now there's a general need or want to look throughout the proteome and trying to understand on really any given protein, but potentially future therapeutic targets, which cysteines are targetable. Now, to do this, there's been a lot of really great work on trying to understand which cysteines on a molecule might be accessible. And a lot of this was done in Ben Kravat's group. I'm going to show you an example a little bit later in a multiplex sense from Steve Geege's group. But these approaches have been really kind of the core functionality of chemoproteomics as a field. Now here what I'm showing is a situation where we're taking cells, we're doing a lysis in a native environment to keep the protein structure. Once we do that, we can do one of two things. We can treat with a compound, or we can just treat with basically a vehicle treatment, or DMSO. Now, if the cysteines on the protein are accessible and the compound, say, binds in that particular pocket and then locks onto the cysteine, we're going to get these molecules that are actually bound now to the compound. And what we do is a second step then of this probe labeling, where we have an iodocidamide probe, in this case with a biotin tag, could be something like desthiobiotine or just biotin, and then you label all the remaining cysteines. So in this case, every cysteine, basically, that was not hit by the drug is now going to have a biotin on it. You can then follow the normal TMT labeling, reduction, alkylation, digestion, TMT labeling, and then do a biotin enrichment. And again, remember, so now any peptide we're pulling down is actually a peptide that was not hit by the drug in the vehicle condition or in the compound condition. So when we look at the quantitation then, what we can understand about the cysteines within each of these proteins is how ligandable it is. So if a cysteine has high ligandability, then the compound would have hit it. So the compound would have blocked it from the iodocidamide biotin tag, which means that it would never be in our sample after enrichment. So in this case, a high ligandability compound will actually have a deficit in the TMT signal because it actually was reacted with the original compound. Any cysteine that's not really ligandable would have actually not been labeled by this compound, would have basically just been labeled by iodocidamide equally between the compound and the DMSO control, so you wouldn't see a difference there. So this is a really powerful approach because it allows you really to look at any cysteine that you can detect within the proteome and understand how ligandable it is for the compounds that you care about. Now, obviously, other folks have done this in a really larger scale fashion. This is a really nice example from Steve Gigi's group, where he takes 285 different electrophiles in this case and does a really high throughput analysis demonstrating that you can get through these in replicates in a short amount of time. He's also had the experimental conditions here or proposed that you might be able to get through thousands of molecules a month. And this has really kind of invigorated the chemistry folks within Genentech and our collaborators, who really now want to take a step back and try to take a more holistic view of which cysteines might be targetable by, say, compounds in our library or compounds in literature for therapeutic development. So because of that, there is kind of this need to analyze more samples and to do this in a more high throughput and a more reproducible manner. So Hannah in the group actually had the idea of applying the hyperplexing method to cysteine profiling. And the idea with hyperplexing, as shown here in Steve's paper with Noah from a little while ago, is that you combine basically, say, SILAC labeling. In this case, you would have light, medium, and heavy SILAC labeling. And you can combine that with TMT. So if you combine that, in this case, with 6-plex TMT and 3-plex SILAC, you get an 18-plex sample. Now, obviously, today we have 18-plex reagents, but you can imagine if you could do this in 18-plex TMT with 3-plex SILAC, you can get up to more than 50 samples in a run. So this particular approach is really attractive for increasing the throughput while also increasing the coverage and the reproducibility and the completeness of your data. But there's always going to be some drawbacks. And the drawback is really twofold. One is that you do increase the complexity of your sample quite a bit, which means that you're ultimately not going to be able to sequence as deep as you would have normally. And that's one challenge. The other challenge is that even though we have all three of these within the sample, even if you can kind of control their relative abundance to each other by basically moving out the replicates and kind of having them mirror each other as much as possible, you don't always see sequencing or quantitation of each of the pairs. So you might only get one or two, or potentially in the best case scenario, get all three sequenced and quantified. And especially for cysteine profiling, where this is happening at the peptide level, we want quantitation at the peptide and site level. We really need quantitation on each of these partners. So we wanted to start to understand within cysteine profiling, again at the peptide level quantitation, how does hyperplexing or what does hyperplexing look like? Now, Hannah came up with two different ways of approaching hyperplexing. We had SILAC TMT, which is kind of the traditional one that I just talked about, where you have SILAC light and heavy cells, and then you can layer TMT on top of those. But within Steve's paper, he actually kind of discretely described a heavy and a light DBIA reagent, which is the desthiobiotine iodocidamide reagent, which is the probe. there's actually a description of that. So we also used this. So in this case, we don't have to grow cells in light or heavy silac media. We can just grow them normally and then label them either with the heavy or light DBIA reagents. So we can get a 2-plex hyperplexing in that case, all the way up to 36 samples in a run. So obviously the labeling is the preferred approach when you don't have to grow the cells in silac media, and you can adapt it more easily to things like tissues if you want to do that. And to analyze this, and this is kind of a key point for any hyperplex sample, Thermo's actually had some experience in this in the past, and they have this really nice targeted mass difference filter within the normal method editor, particularly for things like the Explorers and the Eclipse and all their newer instruments. So with this filter, it allows you basically to tell the instrument how many precursors within the group you have. In this case, we have 2. It also allows you to fill out what the mass differences are of your different labels. In this case, we have lysine and arginine, so 8 and 10. And then you can set things like the mass tolerance. You can set the relative intensity, which in our case should actually be pretty tight, because when we're going to set up our experiments, we're going to have those be mirrored with 2 replicates each. So we should have a total of 4 replicates in our experiment, 2 in each cluster. And then you have the option to perform the subsequent scans, in this case on both ions, right? And we want them to be the same charge state. So in a perfect world, what we would see is that the instrument would find these pairs, these 2 blue pairs, they would identify them as pairs, and then it would sequence both of those. And we'd get 2 MS2s that look almost identical, just shifted over by the mass of the silac reagent. And this is kind of the backbone, because every method that I'll show you within this presentation actually used this targeted mass difference filter to try to sequence pairs as much as possible. So using that, we use that filter in 2 different ways, either without real-time search, so this is SPS-MS3 without real-time search, or we have SPS-MS3 with the real-time search. And the height of the bars here is the total number of unique cysteine-containing peptides. So that's kind of the maximum amount that we identified across both clusters. So that's unique peptide sequences. The darker bar here is basically how many of those were quantified in both of the pairs within each of these runs. And of course, we have silac TMT on the left and heavy light DBIA on the right. So what you can see from this, there's a couple things to take away right away. One is that ThermoRTS, the real-time search through the Thermo method editor, does improve the total number of unique cysteine-containing peptides pretty dramatically. So in that sense, using real-time search really seems to have a benefit. But if you look at the overlap in terms of the percentage of pairs that we're getting, you can see that it's generally going to be anywhere from 64% all the way up to 76%. And interestingly enough, the real-time search seems to actually decrease it somewhat compared to just doing the no real-time search. And that could be because it's going faster and digging deeper into the sample, but it's something that we noticed. Now, one of the things that has been kind of recently described in the field most notably by Bernard Kuster, potentially others, is this idea of kind of transferring quantitation or transferring identifications when you have precursors that might have been selected between 2 runs and you have quantitation on them, but maybe for some reason or the other, one of those peptides didn't pass the false discovery rate threshold. So what we wanted to know here was, well, what happens if we try to look at peptides where one of the partners passed our false discovery rate threshold and the other partner was identified as the correct sequence, but maybe just didn't reach the FDR threshold for some reason? Can we rescue those quantitation events from the ones that might have just been below the false discovery rate cutoff? So if we pull those up, really requiring that at least one pair passed false discovery rate, we can see that the percentages do improve quite a bit. We get now anywhere from 71% all the way up to 83% of the pairs that are totally quantified in both of them. But you can see that there's still a pretty big gap at the top, somewhere around 20% of unique peptide sequences that we've identified just don't have quantitation in both pairs. And that's really the area that we wanted to improve, because we thought that there should be really no reason why that can't be 100%. So what we did was introduce NCKPI. So if you haven't heard us talk about that before, NCKPI is our own internal API program that we have that interfaces with Thermo instruments, in this case with Eclipse or Ascend instruments, and will collect data in real time onto the computer that's associated with the instrument and then analyze it determine what you want to do, something new potentially, and then send those instructions back to the instrument. So in this case, for all of the cysteine profiling, we developed two different methods. One is a real-time search method that's very similar to thermo real-time search, only we included this modification filter, which only quantifies cysteine-containing peptides. Now, for these, it doesn't make too big of a difference because about 90% or more of the peptides contain a cysteine. But for things like phosphorylation and other approaches, this would actually be a really nice filter to have. We also created this pair quant filter, which I'm going to go through right now. So when we looked at the data, those data where we're trying to figure out that missing 20%, we really kind of categorized hyperplexing method acquisition into a few different scenarios. So we have one scenario where we have the kind of the correct scenario, right? We have the pair selection that occurs the way it should. So in this case, we have the pairs that are identified, and then each of those is sequenced, and both of them get identified correctly. So that's kind of the best case scenario. We found some cases which are kind of perplexing at first, where both of the correct peaks were sequenced. In this case, it looks identical to option one, but for whatever reason, one of the peptides didn't actually match to the same peptide sequence. And in some cases, what we found is that this is actually because that peptide sequence was within, say, the top 5 matches to that spectrum, but it just wasn't at the top. So it might have been the second best peptide match to that spectrum, but by looking at them, they look almost identical. We also had cases where maybe the incorrect pair was picked. In this case, you would see that, this was the red and the blue instead of the blue and the blue. In this case, obviously, these would either be two different peptide sequences, both identified, or it could be that you would have one that basically didn't match anything or match a decoy, and one that matched the right peptide. So there could be some issues there. And then the last one was one that was kind of the most interesting for me, was that even though within the targeted mass difference filter, we're asking it to actually give us scans on both of the pairs, and it does those subsequently, one right after the other, typically. We found that there were some cases where only one MS2 was actually sent. And in those cases, obviously, we had no chance of getting the pair. So those would be situations where you would never even have the opportunity to sequence the pair in that sense. So what we did was we took all these cases that we broke these down into these different kind of failure modes. And we asked, well, what happens if we once we identify a peptide, if we don't identify its partner, basically send scans to do that identification. And that was part of this pair quant method. And it really starts from an instrument MS2, which gets put through the pair quant node, which does the identification. And if it's the first time you've seen a peptide, it kind of saves it on a list. And then we have a different node, this pair seq node, that basically will ask, okay, well, if it's been 2 scans since I've seen this peptide and I haven't detected the partner, let me go ahead and send a scan to the instrument to try to identify the partner. And that's what happens here in the middle. So basically, in the cases where we've identified one peptide, we haven't identified the partner, we actively send scans to go sequence the partner. And because these are generally have the same retention time because they're the exact same chemical structure of the TMT tags, and the isotopes from Silac don't actually impart a difference, or from the tag, from the DBIA tag, heavy light, then we have a very good chance of basically, if we've identified the peptide, finding its partner if we just do the right MS2. And that's shown here. So we found that basically in all of these cases, we can send the partner peptide and we can get an identification. And for things like the second one, we were actually surprised that even if we just retake the scan, potentially with a more accurate theoretical mass on the MS2, we do actually get the correct peptide sequence down, particularly in the first position. So that was an interesting one. And then the way that this algorithm also works is that it actually holds back taking the quantitative scans until both of the peptides have been sequenced. So it won't actually send the SBS-MS3 scans for either of the peptides until both have been sequenced. What that means is that if you never actually identify both of the peaks or both of the partners, you won't actually ever take the quantitation, which could be a downside and could lead to a decrease in the overall number of quantitative events, but kind of maximizes pair quantification, which is kind of why we call it pair quant. So to do this comparison correctly, we wanted to not only compare to just hyperplexing the hyperplexing method, we wanted to see what would the comparison be if you did 2 runs. And these are always kind of the hardest comparisons, because especially if you're looking at overall total unique sites that you're able to quantify. So again, we have Silac TMT on the left, we have heavy light DBIA TMT on the right, and here you can see we have two TMT plexes with three different methods. In this case, because they're not hyperplexed, we have SPS MS3 with no real-time search, we have the thermal real-time search, and we have in-seq API real-time search, that other method I talked about. You can see that generally the IDs go up as you kind of go across these methods. In-seq API's real-time search does a little bit better of a job than thermal real-time search, but really not too much better. And you can look again, same trend in terms of the overall paired quantification events. It's about 66% for no real-time search, and or somewhere around 60 to maybe 64% when you have a real-time search employed. Okay, so that's two runs. Now we're going to go to the hyperplex samples. And when you go to the hyperplex samples, again, we have kind of two things to notice here. The first one is obvious what happens with the total number of unique cysteines that you're able to quantify. Now this goes down quite a bit. It goes down from somewhere around 24,000, say, to 16,000 for the pair quant method and in-seq API. But remember, we're only doing one run now. So instead of doing two runs, we're doing one. So we're cutting the acquisition time in half. And what you've probably noticed is that we're actually also greatly increasing the percentage of paired quantification. So for the no real-time search, it's about 80% kind of in both methods. Thermal real-time search, maybe around 75. But with pair quant, it's really between 98 to 100% of paired quantitation events. And that's really coming from the fact that we're waiting until both of the sequences have been associated with a particular MS2 scan before we're doing the quantitation. So it's going to give us this real big boost in terms of complete quantification events. Now, obviously, we're going to lose some on the total number of sites, unique sites that we can get. And that's really just because we have half the instrument time. So, this is kind of where people might have the option. You can get similar types of complete data, which is what you can see here. The complete data bars are pretty close, these dark bars. You can get similar complete data points without getting some of the extra ones that might not have complete data. Or we thought, okay, well, what happens if we take our hyperplex sample, just do two fractions, same amount of instrument time, same amount of data collection time, and now we analyze these, in this case, with two different methods, either thermal RTS or pair quant. And what you can see here is now for Silac TMT, we've actually matched the total number of unique cysteine-containing peptide sequences, but in this case, we have complete coverage. And we're still a little bit lower for the heavy light DBIA. It's still a little bit lower than just doing two complete plexes, but again, we have just way more complete quantification events where we have quantitative data on both hyperplex pairs. So we think that this is like a really nice approach if you're really interested in complete quantitation, particularly here where all of the data is going to be quantified at the site level, which really requires peptide level quantification. So obviously we wanted to use this on a sample, on a particularly interesting kind of profiling example. And Hannah has in the past been really interested in subcellular fractionation, kind of protein communities, how proteins exist in different parts of the cell. So her idea was to do a nuclear preparation first, do a nuclear lysate, and then do this cysteine profiling with a nuclear lysate. And there's a lot of really good precedent or reason to do this. A lot of the proteins that we'd like to build therapeutics for would be transcription factors or other proteins that might exist in the nucleus. And it's possible that the proteins, maybe some of the same proteins that exist in the cytosol might actually inhibit our ability to find some of these unique cysteines that might be uncovered due to things like protein-protein interactions or lack of protein-protein interactions in the nucleus. So we employed this particular approach. Again, in this case, we have two replicates in plex 1, two replicates in plex 2. All of this was combined, hyperplexed together. And you can see that overall, in terms of cysteine sites, we got about 10,000. We got about 97% or so complete pairs. And we know that that lack, that 3.3% that's missing, really came from low signal in one of the quantitation events. And that's something that we kind of knew might happen, particularly in a real sample like this, but something that doesn't bother us too much at this moment. Now you might be wondering, how does the quantitation compare, particularly across the two different plexes? So in this case, they have the heavy DBIA labeled on the x-axis, light DBIA on the y-axis. You can see in general the quantitation between these two, in this case, so the compound over DMSO, agrees pretty great in terms of the agreement here. The R squares are pretty nice, but there's one that's not. So there's definitely something that happened with one of these replicates, one of these samples, that's causing one of those to not behave quite well. So overall, we're pretty happy with these results, but obviously, potentially, you're always going to be subject to potentially one of your samples causing an issue. This is the kind of the example data for one of these sites that we found that was specific to the nucleus. In this case, it was HES1, the cysteine 128. This is present on the orange domain, which is potentially involved in its DNA binding function. Shown here, you can see the MS1, this is the light, and then the heavy DBIA separated both by here. This is a charge state 3. These are the two MS2s. And then remember, we're doing SPS MS3, so we're selecting from these B and Y ions to get more accurate quantification, particularly in situations where the spectrum might be a little bit more noisy. And you can see here's the heavy, the light DBIA quantitation and the heavy DBI quantitation, very similar in terms of the quantitative value between the two clusters. This is the overall data. I won't really harp on this too much. We do have a bioarchive coming soon and then a submission. But here we compared it to whole cell lysate. So we looked at the nuclear lysate versus the whole cell lysate, cysteine profiling in both. All of these sites were found unique within the nuclear lysate. And you can see that there's a lot of proteins where once you go into the nuclear lysate, you can start to uncover cysteine sites that potentially you hadn't detected before. On things like on COL2, we start to see a site we hadn't seen before. So I think, particularly for higher throughput analysis, if you're okay potentially with getting fewer total sites than you might in again, doing 2 different runs, but you want more complete data all together in one run, I think this is a really nice approach for that. And that data did get us pretty excited to move into a different aspect. So we really wanted to take that data and to move into single cell proteomics, to take this method and to move into single cell proteomics. Now, obviously not the first to think about this. You're going to hear from Ryan a little bit later, Ryan Kelly, about his hyper-SCP method. So the hyper-SCP method in this case is the same idea where he's going to be combining SILAC, light and heavy cells, together with TMT. And he'll describe this a lot more. He obviously, in this case, created a new chip to do this, was able to sort the cells, heavy and light, into these, and then label with TMT as they normally would in terms of a nanopots preparation, and then do a very similar hyperplexed analysis as I just described. Now, not to spoil his talk, but I do want to kind of go over the high-level results from his work. And in terms of protein groups he was able to identify without a carrier proteome, it was somewhere below 500 proteins he was able to quantify. If you look at a 10 nanogram carrier proteome at 90 minutes, he's somewhere closer to maybe 1,000 proteins, and at 60 minutes closer maybe to 800 proteins. And that's something to remember because all the data I'll show you is going to be on this 90-minute gradient, which is much closer to his 1,000 protein level. And then you can see here he has about 620 proteins where he's able to do some differential analysis. So obviously when we started talking about this with Ryan had presented this a little while ago at some conferences, we were excited to kind of take that idea and to apply the pair quant method. So to do that, Tommy Chung and the group kind of led this effort internally, and Ying helped him out with some of the nanopots sample preparation approaches as well. So in our case, we're going to have light HEC293 cells, heavy HEC293 cells, light HeLa cells, and heavy HeLa cells. And we're going to be sorting those cells into an N2 chip. Now, an N2 chip only has 16 spots, and we're going to sort 14 different cells into those spots. But for our purposes, we actually thought it would be a little bit easier just to sort the light 293 cells first, and then say sort the heavy 293 cells right on top of those. Same thing with the HeLas, light HeLas first, heavy HeLas right on top. So in this case, each of the nanopots droplets actually has 2 cells rather than just 1 cell. In addition to that, we used the regular nanopots chip to prepare carrier proteomes that had 40 cells per hyperplexed partner. So we actually sorted 80 cells total into each of these spots, each of these nanopots. So wells, we actually sorted 80 cells each, but in each of those it was 40 light and 40 heavy, and then it was 20 light 293, 20 heavy 293, 20 light HeLa, 20 heavy HeLa. But overall, the effective carrier proteome should be around 40 times carrier proteome. Then the sample preparation was done separately on both of these chips, and at the end of the day, we combined the wells from the carrier with one of the wells from the N2 chip, and then analyzed it with paraquant on our Orbitrap clips. All of these were done in 2-hour runs, which included a 90-minute gradient. So we think that could be decreased somewhat, but we were just kind of being cautious for these first runs. Obviously, we use the paraquant method shown here, where we're going to be waiting to identify both the pairs to send. In this case, FTMS2s. We're not doing SPS MS3 at this point. We're doing FTMS2. So these are the 4 different methods that we used for this particular example. You can see here that we have kind of the control method, which is just a thermo FTMS2 method. No real-time search here, because when we tried to load that database on to do the real-time search on Thermo's instrument, it actually didn't work. So we're just going to do straight up FTMS2, no real-time search. We are using the targeted mass difference filter here. Then we have a real-time search that's really similar to what Erwin Schuf would call the reticle method. In this case, we're also using targeted mass difference. We're triggering ion trap MS2 scans, but then we're using InSeq API in this case to do a real-time search, applying what we're calling protein closeout, which I'll talk about in a second, and then we're triggering an FTMS2. We also have paraquant, and in this case, it's the same paraquant method that I've just described to you already. However, we have that with and without protein closeout, which again, I'll talk about. So we have 4 different methods that we use to analyze these to try to understand how they're going to fare. So I do want to talk very briefly about protein closeout. So protein closeout is a feature that's already available on Thermo's real-time search, and allows you to basically keep track of which peptides you found and which proteins they're associated with. And once you found so many peptides per protein, say 3 peptides per protein, you can stop collecting peptides, quantitative data on peptides for that protein. Now, the goal here is so that you can dig deeper into the proteome, specifically for complex samples, so you don't keep collecting, say, data on actin or keratin, something where you don't want to just keep collecting peptides over and over and over again. One of the challenges with doing this type of approach, particularly in real time, is how you deal with shared peptides. So currently, the way that shared peptides work within the implementation on the instruments is that shared peptides will go to the first entry into the database. Now, the challenge with this is that if you happen to get 3 shared peptides first, in this case between protein A and protein B, and you only wanted to do 3 peptides total, you would actually potentially close out protein A before you got to other peptides. Now, the area where this would be a challenge is if perhaps after these first 3 shared peptides, you started collecting distinct peptides. And maybe there was an option to collect this distinct peptide A1, distinct peptide B1, and B2. Now, B1 and B2 would be collected, no biggie, because there's no peptides on that list. But if you closed it after 3 peptides, you would actually close protein A on the 3rd shared peptide. In that case, you would never actually be able to detect or get quantitative information on A1, the distinct peptide. So what we did with an InSeq API was actually change this up a little bit. We put, in our case, 3 different lists: distinct peptides, which belong to only one protein within the database. That's one peptide to one protein relationship. Unique peptides, which is just a running list of any peptide you've quantified for that particular protein. And then obviously PSMs, which is how many times you've hit any peptide for that protein. And for our list, we actually allow you to set a unique peptide threshold, which in this case, if you hit 3 shared peptides first, those 3 shared peptides would go into the list I've shown here, both the unique list as well as the PSM list, but you would stop collecting shared peptides at that point. So if another shared peptide came in for A or B, wouldn't do it. Even if there was a shared peptide between B and C, it wouldn't actually take that peptide because it kind of assumes that you've had enough shared peptides with B. But what it does allow you to do is actually continue to collect distinct peptides. So in this case, if the distinct A1 came along, you would still be able to collect it until you get to a distinct peptide threshold. In this case, say it's also 3. Same thing with distinct B2. So you would have a threshold that you might want to have 3 distinct peptides, 3 unique peptides. If you hit 3 distinct peptides first, those actually fill up the unique peptide spots. But if you hit 3 shared peptides first, it doesn't fill up the distinct spots. So in that way, you should actually be able to keep quantifying distinct peptides if you don't think you've had enough. And I think this is one of the areas where some of the results I'll show you, we have pretty good coverage in terms of the protein numbers, and I think that might be why. So the very first thing we typically do when we look at our methods that we're trying to assess is look at how much signal we're getting and the estimated carrier ratio for using carrier proteome. In this case, you can see that the thermo method, the FTMS2 method, is not quite giving us enough signal that we might like. The red line is where we're hoping it would be. For any of the real-time search methods, either API real-time search or both of the pair quant methods, it is. So there we're having a lot more signal, particularly in the pair quant with the protein closed, which was somewhat interesting. So for the estimated carrier ratio, obviously we put it at 40. We'd like to see it at 40. As you can see here, most of them are around 45-ish. So that's not too bad. That's around where we would think it is. And the fact that it's not exactly 40 might be just due to lower signal. But you can see here, pair quant with the protein closed right at 40. So we're very confident in terms of the data quality, particularly for the pair quant data that we're able to have. So the super high level results, looking at identified proteins, proteins with quantitation, and then proteins that we actually called quantified. So identified proteins are ones that where we just had an MS2 that was identified to a particular sequence. Proteins with quant means that we actually triggered an FTMS2. Now, remember, that won't always happen, particularly with protein closeout. There's going to be a lot that if the protein is already closed out that we're not going to be triggering FTMS2 on, so those bars will be potentially a little bit lower. Then we have quantified proteins, which means that we've applied our kind of stringent, actually pretty stringent cutoff for the signal. We have, in this case, I think the signal to noise cutoff was like 220. I'm sure a lot of other folks in the field use something like 100. So it's a pretty stringent quantitative cutoff at that point. So in this case, you can see that thermo FTMS2, once you go to quantified peptides, it's really down to about 500 proteins. So that's built off of peptides that were basically quantified to that level. And then with no protein closeout, in this case, with pair quant, you can see it's a little bit higher at 600. Obviously, the protein closeout methods are giving us the highest numbers here, either API real-time search or for pair quant, they're also somewhere around 1,000. So this is just how many proteins we're able to quantify if we just kind of ignore the pairs that we wanted to quantify. So now we want to look at the pair quantification, right? Because that's what we're really trying to understand here is what happens if we build off of data where the pairs were complete at the peptide level. So to do this, I looked at the peptide level and I asked, okay, for the peptide, did we quantify both the light and the heavy? And remember, we're applying those quantitative thresholds to both of them that are pretty strict. So this is the number of proteins that were built off complete peptide data. And this is per run number of proteins. So for thermo FTMS2, we're getting about 300 peptide or proteins quantified off of complete data, and for pair quant without protein closeout, it's closer to about 500 or so proteins. For the real-time search with protein closeout, it's close to about 600, which is very close, which is about 40% down in terms of how many we were able to identify. But if we look at pair quant with protein close, we're at about 950 proteins that were built off of complete data within each run. And that was pretty exciting. So we're getting very close to really what someone like Ryan had published for the same type of gradient length. But here we're getting really complete data at the peptide level, which we're building these proteins off of. So that was really exciting for us. If you look at the percentage, right, now, again, we have the initial and then after the FDR rescue here, as we did on the last slide. If you look at the percentage, you can see really the real-time search, again, somewhere around 60%, which is kind of what we saw in the cysteine profiling. For thermo with the FTMS2, it's going to be a little bit higher. It's about 65% after rescue. But for pair quant, both of the methods, really, irregardless of doing protein closeout or not doing protein closeout, you can see that it's about 85 to 87%. And that's already exciting, but again, kind of being perfectionists, we're still looking into how we can bump this up. We'd like that to be closer to 90 to 95 to, or even 100%. We really don't think there's too many reasons why we shouldn't be able to basically quantify both of the pairs within a single cell proteomics experiment. And a lot of these might be due to the differences between FDR on and offline or signal to noise filtering, things like that might cause this difference, but we're looking into that now. But overall, super happy with how that turned out, particularly the depth that we're able to get at while having this hyperplex pair. So if you look at the total number of identified and quantified proteins across all of the runs, so this wasn't too many runs. For the protein closed out in pair quant, it was 3 separate analyses. So that was 3, in this case, it was 14 cells in each hyperplex pair, so it's 3 times 28. So we were able to do about 84 cells in 3 runs for pair quant protein closeout. For thermo FTMS2, obviously this was 3 runs. For the other 2, it was only 2 runs. So you can see that the number of proteins goes anywhere from 500 to about 650 with pair quant and no protein closeout, about 800 with real-time search and protein closeout, and about 1,300 proteins we were able to get in any of across all of the runs with pair quant and protein closeout. So that was particularly interesting. And then, of course, we can look at the data. So this is taking data that was complete across all 84 cells for the pair quant with protein closeout. I believe it's about 650 or 700 proteins that were quantified across all. And we're not really doing anything fancy here. You know, some very light normalization, which we found still had a lot of batch effects within it. But you can see here we have all of the cells. Generally, they're clustering within the cell type. We have one over here, 293 cell that doesn't. But we do see some batch effects, particularly with 293, where we get kind of all of plex 2, 1, and 3 together, and then even some heavy and light clustering. So we're going to be kind of taking this data, trying to apply some more normalization to it, some batch corrections. But super excited about this because we are able to get, in this case, it was a 2-hour run. We're able to get 84 cells in at a time, and we do think we'd hopefully be able to decrease that runtime with some different intelligent data acquisition approaches too. If you're interested in knowing without the carrier, just really briefly, if you look without the carrier, you actually see that this is the total number of identified and quantified proteins. The real-time search with protein closeout versus pair quant with protein closeout. Real-time search gets about 790 proteins. Without the carrier, pair quant gets about 719. And then if we look to complete pairs again, so this is proteins built off of complete pair data, for a regular real-time search, something like the reticle method, you should get about 480 or so, and for pair quant with protein closeout, closer to 600. So, again, comparing the 600 number to the 950 that you would get with a 40x carrier still seems like it's beneficial to use the carrier proteome, but something to keep in mind, especially as the number of plexes goes up, maybe the reliance on the carrier might start to go away, and we might be able to get pretty good data with even higher throughput. So with that, I hope I've kind of introduced maybe some of you for the first time to this idea of hyperplexing. Set it up for Ryan this afternoon. But if you already knew about that, hopefully I've shown you through both the hyperplexing on cysteine profiling as well as single cell proteomics that there are some shortcomings to the methods that might be available. But through intelligent data acquisition, we can actually improve this through the cysteine profiling anywhere between 96 to 100% of complete pairs, and for single cell proteomics above 85% currently, and hopefully get that up higher, and that we are able to analyze a higher number of cells in a more quick manner. With that, I'd like to thank the folks that did the work on this. Obviously, Tommy and Ying, who worked on the single cell proteomics experiments, and then Hannah, Taylor, and Nina, who worked on the cysteine profiling, hyperplexing, all of MPL. I'd like to thank you for your time, and then leave you with this short disclaimer about some of the talk and Thermo's support of it. So I'd like to thank you again, and happy to answer questions during the roundtable that's going to be upcoming.